Good afternoon, everybody. Thank you so much for joining. We will just give another second or two for more people to filter in and then get started. All right, we can go ahead and begin. Good afternoon. Thank you so much for joining today's webinar, co-hosted by the Hepatitis B Foundation and the National Viral Hepatitis Roundtable. My name is Beatrice Sovich, and I am a Public Health Program Manager at the Hepatitis B Foundation and the manager of our Hepatitis Delta Connect program. I'm joined today by my colleague, Daniel Raymond, who is the Director of Policy for the National Viral Hepatitis Roundtable. Thank you all so much for tuning in today. Today's webinar will provide an overview of the clinical trial process, current hepatitis B and Delta therapies, and an introduction to interpreting clinical trial data. This webinar is part one of a two-part series intended for patients, advocates, and other stakeholders who are interested in learning more about the clinical trial process for hepatitis B and Delta in order to prepare for and engage in the upcoming annual meeting of the American Association for the Study of Liver Disease or other advocacy opportunities in the future. We are thrilled to be joined by three experts in the field who will each address one of these issues. We are looking forward to hearing their insights. Next slide, please. Thank you. So before we get started, I would just like to remind all participants that during today's webinar, all audience members are muted. We encourage you to use the chat feature to let us know who you are and where you are joining from. If you have any technical challenges, please indicate your issue using the chat box and we will address it as soon as possible. If at any time during the presentation you have questions, you can feel free to enter them into the Q&A box. Questions will be addressed by the speakers at the conclusion of the webinar. This session is also being recorded and slide presentations will be shared following the webinar. Next slide, please. Thank you. So just to provide a brief overview of the Hepatitis B Foundation. So we are the only national nonprofit organization that is dedicated exclusively to finding a cure for and improving the quality of life for those affected by hepatitis B around the world. And our commitment includes a number of different aims, including funding focused research, promoting disease awareness, supporting immunization and treatment initiatives, and serving as the main source of information for individuals with lived experience of hepatitis B and Delta and their families and caregivers, the medical and scientific community, and the general public. We also manage focused programming around hepatitis Delta awareness and support, and lead coalitions to increase hepatitis B awareness, screening, vaccination, and linkage to care among highly impacted communities, and work to resolve disparities in liver cancer through education and culturally appropriate communication. Next slide, please. Thank you. And then to provide a quick overview of the National Viral Hepatitis Roundtable. So NVHR is a coalition of patients, healthcare providers, community-based organizations, and public health partners who are working to fight for an equitable world that is free of viral hepatitis. NVHR aims to support a comprehensive and integrated approach to viral hepatitis by centering the voices of those with lived experience, fighting for equitable access to viral hepatitis prevention and care, and advocating for fully funded and robust state and federal elimination plans. Next slide, please. Thank you. So now to begin introductions of our speakers. So we're very excited and honored to be joined today by three outstanding guests. We will first hear from Dr. Christine Lee, who is a Strategic Research Engagement Lead at the Office of Minority Health and Health Equity and the Office of the Commissioner at the U.S. Food and Drug Administration. In this role, Dr. Lee leads minority health and health disparity-focused research and develops strategic partnerships to advance the health of diverse populations. Today, we are looking forward to hearing her present on research and innovation in clinical trials, collaborations and partnerships in the public health space, and how these relate to health equity. So Dr. Lee, I will now hand it over to you. Thank you so much. I think you can stop sharing this screen. Thank you.
Thank you so much, Beatrice. Let me go ahead and share my screen. All right. So good afternoon, everyone, or good morning. It's a delight to be here today. I'm going to be talking a little bit about the work the FDA Office of Minority Health and Health Equity does in advancing racial and ethnic minority participation in clinical trials. This is our standard disclaimer slide. So a little bit about the FDA Office of Minority Health and Health Equity. Our mission is to promote and protect the health of diverse populations through research and communication that addresses health disparities. Our vision is one, to create a world where health equity is a reality for all. So what do we do within the Office of Minority Health and Health Equity? We have two programmatic areas, and I will go into both of the programmatic areas in more details in the next slides. The first program that we will talk about today is research and collaboration. Within this program arm, we fund intramural research and extramural research, as well as we work through our Centers of Excellence and Regulatory Science and Innovation called Cirrhosis. We also leverage mechanisms such as the broad agency announcement, and we also have additional other research opportunities, which I will go into in the next couple of slides. We also fund internships and fellowships, especially with our partners, our National Institute of Health and HGRI. Additionally, the other programmatic area that we have is outreach and communication. Within this program arm, culturally and linguistically tailored programs, initiatives, and campaigns are developed. Health education materials are supported. We put on a monthly webinar series and health lecture series, as well as we host stakeholders meetings, symposiums, and exhibits. Within the communication program, a major part of that program is diversity in clinical trial campaign, in which videos, newsletters, e-alerts, web pages, podcasts are all supported on our web pages. For example, here are some of the examples on our web pages that highlight the videos, podcasts, and social media outlets. Additionally, we have a various multitude of clinical trial diversity resources, including fact sheets and our updated web pages. Now, delving into the other program, research and collaboration, I'm going to go into more details of what this program entails. For example, I had mentioned fellowships. We currently have an OMHHE and NHGRI postdoctoral fellowship in genomics, science, and health equity. We also work with our federal partners, such as the VA, through IAA's interagency agreements. For example, right now we have one that's looking into real-world evidence and artificial intelligence options. We also, for example, work with the Hepatitis B Foundation through an RCA research collaborative agreement, which I will also highlight one of the work products that came out of this partnership. Additionally, we leverage mechanisms such as broad agency announcements, BAAs, Cirrhosis, and workshops. As you can see, we have multiple bridges for us to connect to our external partners. Going into our newest initiative, the Enhanced Equity Initiative. This initiative really highlights the FDA Office of Minority Health and Health Equity research projects and communication resources to really enhance three specific pillars. The first pillar is equity in clinical trials, including supporting efforts to advance diversity in clinical trials. We also have our equity in data efforts, which advances and increases data available to diverse groups, including but not limited to ethnicity, race, age, disability, and geography. Also, my favorite, equity of voices, amplifying FDA's communication with diverse groups to ensure stakeholders, including consumers, are informed about FDA efforts, and as well as to understand diverse patient perspective, preferences, and unmet needs. So here is just a couple of research projects that we work in within the equity in clinical trial space. For example, working with Fullerton University, understanding how to create digital tools to enhance young adult minority participation in COVID-19 trials. We also, for example, work with Howard University to understand the different attitudes, preferences, unmet needs of both the African American and Asian American populations when it comes to clinical trials. As I mentioned a little bit earlier, our RCA with the Hepatitis B Foundation, understanding barriers to hepatitis B clinical trials. And as you can see, we have a quite a long list of diverse partners looking into different subgroups to understand their barriers, preferences, specific unmet needs. As promised, here's a recent publication that was developed through the RCA with the Hepatitis B Foundation, in which we looked at attitudes of diverse participants towards clinical trials with those living with chronic hepatitis B. Going into our equity of data efforts, some examples include leveraging real-world data and real-world evidence among COVID-19 treatment outcomes among racial and ethnic minority groups with their partnership with the Veterans Affairs Administration. Additionally, our current fellow with NHGRI is looking into sickle cell data elements. Additional work includes understanding racial and ethnic differences when it comes to asthma biologics among pediatric populations. And finally, our equity of voices work. As you can see here, many of these projects look to elevate and amplify the voices of populations we typically do not hear from, including older racial and ethnic minorities, including understanding populations from Hawaii, Puerto Rico, and other parts of our country, and as well as leveraging big data, machine learning, and other approaches to advance our understanding of our stakeholders. This is our current fellow between OMHHE and NHGRI. Dr. Raven Harding is a Genomic Science and Health Equity Postdoctoral Fellow. She's conducting research on sickle cell disease, a condition that primarily impacts Black and African American populations. And as promised, we do annually from the office put out an RFA, a UO1 grant. If you're interested, please let me know and I can send you the link. And that is all. Thank you.
Thank you so much, Dr. Lee. We really appreciate your presentation and all of the really important work that you're doing around clinical trial and data equity and capturing unique voices that aren't typically heard. So thank you so much. And we will now progress to our second speaker. So we would like to introduce Dr. John Tavis, who is a professor of molecular virology at the St. Louis University School of Medicine and is the director of the St. Louis University Institute for Drug and Biotherapeutic Innovation. He's also a member of the Scientific and Medical Advisory Board for the Hepatitis B Foundation and the Baruch S. Blumberg Institute. And he's done a lot of work around studying hepatitis B replication mechanisms, currently focused on the basic biochemistry of HBV ribonuclease H and developing drugs to suppress HBV replication that target this enzyme. So today we will be hearing from Dr. Tavis on currently available hepatitis B and Delta therapies and those in clinical trials. So Dr. Tavis, I will now hand it over to you. Thank you so much.
Thank you very much. And we got to do the share screen game now. So here we go. Tell me if you're seeing my screen properly.
Yep.
Okay.
Good.
All righty. So let's get started here. Okay, so I've gotten an introduction, so let's move forward here. My goal here is to give a foundation for understanding current and developmental drugs that are appropriate for hepatitis B and hepatitis delta viruses. So the best place to start whenever you're doing something like that is a real quick virology overview, because I know not everybody has spent 30+ years studying fatal human viruses like I have. So let's get into the viruses just a little bit. First off, hepatitis B virus is a partially double-stranded DNA virus. It replicates in liver cells using a process called reverse transcription that copies a version of the viral genome that is in a molecular type called RNA back into a type called DNA that you're probably a little bit more familiar with. The virus chronically infects somewhere around 300 million people worldwide. These numbers are always a little bit mushy with a worldwide pathogen like this, but that's close to the best current estimates. It causes acute and chronic hepatitis, and that hepatitis in many patients can proceed to cirrhosis, liver failure, and eventually liver cancer in some patients. Together, this causes a little bit less than about 1 million deaths annually, according to the best currently available estimates. In contrast, hepatitis delta virus is a very, very different beast. It is a circular RNA viroid, so it's not even technically a virus. For those of us who are virus nerds, it's not even technically a virus, it's a viroid. It replicates in the liver using human enzymes, and it needs one of the HBV's enzymes called HBsAg, or the surface antigen for hepatitis B virus, to be able to spread between cells, but it can replicate within a liver cell all by itself without any troubles. It is always found together with HBV because it needs the HBV surface antigen to spread. And around 5% of people with chronic HBV also have chronic HDV. However, the diagnostic tests for HDV are not really all that good, and the effort gone into worldwide surveillance is not super high for hepatitis delta virus. So there is a greater degree of uncertainty on that number than we really like to have. And in general, it makes hepatitis worse in people who are co-infected with the two viruses. So what's up for the current therapies that we have for them? So we do have some therapies, so we're not completely depressed. You have, for hepatitis B virus, the first one approved was Interferon Alpha, and that was subsequently modified to a pegylated version that lasts longer in the body. That triggers host innate immune responses to attack HBV. So basically it tells your body, hey, you're infected, go beat up on this virus. And so that's effective in many cases, but the side effects can be serious with interferon alpha in many patients. And it produces long-term liver health improvements in up to 30% of patients who are given this treatment that lasts about a year. However, it very rarely cures hepatitis B virus infections from the patients. The dominant drug currently for HBV, or the drug class actually, is nucleoside analogues, for example, entecavir or tenofovir. These block viral DNA replication, that reverse transcription process I mentioned. They are usually very well tolerated in patients, and they are generally taken for the full life of the person. They improve liver health in almost all patients. The liver enzyme function normalizes in almost everybody. So they're actually fairly good drugs at reducing disease, but they rarely cure HBV at all. The best numbers are between 5 and maybe 7% of patients after multiple years end up being cured of their HBV infection from it. So on hepatitis delta virus, the news is not quite as good. You also have pegylated interferon alpha here, but you have to give it for a longer period of time and a higher concentration. Again, it triggers host innate immune responses to attack both HDV and HBV in the person. But because of the long-term treatment and the higher concentration of it used, the side effects can oftentimes be really fairly severe with this, and many patients can't tolerate it for the duration that's needed. And again, this very rarely cures hepatitis delta virus. However, fortunately, we've just got a new drug that's been approved in Europe and is in phase 3 trials in the United States called Miracledex B. It's also called Hepcludex, and it's got another name that I always keep forgetting, starts with a B. This is an entry inhibitor. It blocks the ability of hepatitis B virus and hepatitis delta virus to get into cells. So it doesn't do anything to the cells that are already infected, but it keeps additional cells in your body from becoming infected. It is usually extremely well tolerated in patients, and it improves liver health in the hepatitis delta virus co-infected patients, but again, it rarely cures the infection. And so we're looking at a glass half full or a glass half empty issue here, where we've got some drugs that help, but they don't go all the way to where we want them to be with the patients. So what are the goals of therapy for HBV and HDV? Well, the general goals are pretty obvious. You always want to stop and hopefully reverse the disease progression in the individuals. That's your big goal right there. Secondary goals is, of course, you'd really like not to transmit the virus to other people so they don't end up with a health problem. And you really want to cure HBV infection so the patients don't have to take drugs their entire life. Now, When you get into HBV here, then you immediately start talking about an awful lot of different names, terminology, and stuff like that. But basically where we're at in the field of HBV drug discovery is we want to achieve something called a functional cure. Now, the biological definition of this, and this is what we want to get done, is you want to have no replicating HBV in the liver or HBV DNA in the blood after the drugs are stopped. That would result in no disease progression and eventually immune control of any residual HBV that happens to be remaining within your body. But of course, that's not something your doctor can measure in the exam room. And so there's also a clinical definition of a functional cure, and that is currently defined as undetectable HBV DNA and HBV surface antigen in blood 6 months post-treatment termination. Now, nobody likes the name of the functional cure name, and nobody likes the clinical definition of it because there's lots of problems with it. Unfortunately, that's the best we're able to do right now, and there's a very large amount of work going into trying to improve the diagnostics around defining a functional cure. So HBV, let's think about drug discovery, because of course we're also looking towards what's in the pipeline available for folks. So what drug targets are available to patients or to the drug developers here? At this level, we're talking about the drug developers. And you can see I've got the cellular replication cycle of the virus laid out here, showing the virus coming in and replicating, comes out and then you get more of it. So in any case, there's no point in going through this in detail because it's extremely busy, and that's its entire point, is we have an embarrassment of riches of targets to hit with hepatitis B virus. The virus is unusual compared to what goes on in your cell on a normal daily basis. Its replication process is complicated. There's a whole bunch of different things that nerds like me in the lab can attack to stop it. And so these are the things that are available to us. That doesn't mean they're all being attacked right now, or they all are, plausible to end up being drugs. But these are the things that you have to choose from when it comes time to start hitting the virus. Same slide for hepatitis delta virus is a lot simpler. And that's because hepatitis delta virus is very simple itself. For example, it doesn't have any viral enzymes. That's a protein that does a chemical reaction in the cell. It doesn't even have any of those. So it's very simple coming in there. So consequently, we've got fewer places where we can whack it over the head. But here again, it shows you a number of different things where we could possibly attack it. And some of these involve immune-related activities here with therapeutic vaccination or T-cell activation, and some involve directly attacking the virus here. For example, the HDV antigen prenylation. I won't get into the details on what that means. It's just that is an area where it's an essential activity of the virus, and there are drugs being developed to terminate it. The other approach is, of course, just go cure HBV. And that would stop viral spread between the cells, and it would eventually clear hepatitis delta virus as the infected cells are killed by the immune system and replaced by uninfected cells. And so we've got two ways to attack hepatitis delta virus. One is attack the virus itself. The other is attack its partner, hepatitis B virus. Okay. So I get to ask this question a lot. I know the folks at the Hepatitis B Foundation get asked this one all the time, is why don't we have a cure yet? I mean, everybody wants a cure. Why don't we have one? So this breaks down into biological and non-biological reasons. So for HBV, the biological reasons are there's a form of the viral DNA called the cccDNA that hangs out in a person's liver. It's very stable, and it is not really doing anything. It's just sitting there. It is being transcribed, it's making RNAs and things like that. But as far as it doesn't copy itself, it just sits there. And it's very hard to disrupt something that's sitting in the nucleus of your cell, just hanging out. And so it's very stable and hard to hit. Second, the virus has a high replication level. And the higher the replication level, of course, the bigger problem you have getting rid of a virus. And then the virus induces immune tolerance to the HBV proteins. Immune tolerance means the the person's immune system, their antibodies, their white blood cells, get kind of used to having the proteins there in one way or the other. And there's all sorts of words involved with this, but it boils down to the fact that the immune system isn't effective in clearing the infection. So the non-biological reasons are, and this is a big one, is underinvestment in research until very recently. I've been studying this virus for 30 years, and it's only been in the last 5 years or so that there's been a significant investment in cure research. That has led to a lack of basic knowledge about how the cccDNA functions and how it's made and how it gets broken down, and also a lack of basic knowledge about the immune system's problems during chronic infection. And so those things together are why we don't have a cure for HBV yet. For HDV, they break down into two groups also. First off is, again, is an extraordinarily high viral replication level. Very few viral targets to attack, which I've described to you in the preceding slide or two. And again, there's poor immune responses to the only protein the virus makes, which is called HDV for the hepatitis delta antigen. So non-biological reasons, persistent and severe underinvestment in cure research. HDV has gotten even less attention from the funding agencies worldwide. It's not just an American problem, it's a worldwide problem than HBV did. There's a lack of basic knowledge about HDV RNA metabolism, how it is made, get turned over, and those types of things that we need to know how to whack it. And there's a lack of basic knowledge about the immune dysfunction to hepatitis delta virus too. And so the theme here is that underinvestment in hepatitis delta virus and HBV research over decades has put us in a hole, and it's a problem we're trying to overcome right now. Okay, so I promised I'd tell you a little bit about the different types of drugs that are available for HBV and what's coming down the pike, and that's what this slide is for hepatitis B virus. Note that things labeled in blue are things that are in clinical trials, and things that are green are things that the FDA has already approved. I'm going to talk about our drug classes here, not any individual molecule. But there's a great reference site that lists the individual molecule that's run by the Hepatitis B Foundation. And the website link is down there. I strongly encourage you guys to go to it because I use it routinely. So again, in the center, we've got the viral replication cycle showing the virus busy doing its stuff within a cell. And so the approved drugs are reverse transcriptase inhibitors. These are the nucleoside analogues that block viral DNA replication. You also have interferon alpha, the type that's used usually these days, the pegylated version of it, and that triggers the immune system. There's a whole bunch of systems within your cell that are capable of digging a virus out of there. And so interferon alpha turns some of those on. Then you've got immune-related activities where there's processes going on to try and directly activate T cells. These are a major class, your white blood cells, to go and attack the infected cells. There's things called TLRs or toll-like receptors on the surface of the cells that act kind of like triggering interferon alpha by turning on the innate immune system to attack the virus. We've got therapeutic vaccinations. These are most of the vaccines you guys are thinking about on a daily basis. Those are prophylactic vaccines designed to keep you from getting an infection. A therapeutic vaccination is a vaccination designed to help cure an infection. And so here you see there's therapeutic vaccination things in trials right now. There's monoclonal antibodies. You probably heard about some of those for COVID earlier in the pandemic. So monoclonal antibodies are being developed. The leading class of compounds is probably these capsid stability modulators, are called CPAMs, that break down the protein shell that surrounds the virus. And then you've got a class of compounds that inhibit surface antigen release, and it also inhibits viral release. And then the entry inhibitors are there. And RNA stability, these are siRNAs and antisense RNAs designed to destroy the viral RNAs. So there's a whole bunch of stuff going on with multiple drug candidates in each of these categories there. And again, more information is found here at the Hep B Foundation's website. Same type of slide for hepatitis delta right now is, again, green is approved drugs and blue is in clinical trials. The entry one listed up here is a little bit misleading because it's in clinical trials in the United States, but it's been approved in Europe. So it's crossing that line right now, and hopefully it will eventually be approved in the United States also. And then you've got interferon alpha that I described earlier. The experimental drugs, you have interferon lambda, which is a safer version of interferon alpha. It works in at least conceptually the same way, but it tends to be a little bit easier on patients. You've got these prenylation inhibitors coming along that stop viral replication. Those are in clinical trials. You've got some drugs that are working to try and block release of the hepatitis delta virus, and of course the entry inhibitor that I mentioned. And of course, HBV targeting mechanisms is really where most of the effort is going on here, because if you get rid of B, you get rid of delta. And here is HBV antigen release. Of course, I made a mistake, and that's on there twice. Sorry about that. You got monoclonal antibodies and RNA stability. And again, the reference site here, it's got both delta and B virus data in it. Great site. Okay, getting close to the end here. So the hurdles to expanding treatment indications for HBV and HDV therapies. This is a question the foundation gets fairly often is, okay, why can't we, start giving the nucleoside analogues earlier? Because it's a very complicated algorithm indicating when it is time to start giving drugs to the individuals. And so there's a number of things to keep in mind when you're doing this so that you can understand the constrictions that the drug companies and the FDA are facing as they try and navigate this very complicated topic. Is first off, HBV is a very slow disease course. It takes decades. Typically, if you're infected at birth, 5 or 6 decades before you start reaching the traditional clinical endpoints of the terminal end of a disease point, which for HBV would be like developing cirrhosis or cancer onset or something like that. And it is simply impossible to design a clinical trial to assess those endpoints directly. So you need to use something called surrogate endpoints to measure these things like viral reduction in ALT levels and things like that. But of course, those don't necessarily track perfectly with the end point that you want to measure. And so that just adds complexity to the whole process. Second, and this is kind of counterintuitive, we have safe and partially effective existing drugs. The nucleoside analogues do a pretty good job at suppressing primary disease with a hepatitis B virus. And so the new drugs, by definition, must be non-inferior. That means they got to be as good or better than the existing ones in order to reach the FDA's guidelines and definitions for approval of a new drug. You also have to really watch out for safety. And so the new drugs cannot be more dangerous than the existing ones unless there's a really, really clear reason why to use them and a way to manage that danger. And that makes designing clinical trials difficult, as the FDA is naturally intent on only permitting minimal risk under those circumstances. Remember, one of the FDA's primary rules, and probably its single most important central rule, is to protect the American population. And so they are concerned about risk under these circumstances. And the final thing that makes it a little bit different is the conservative medical guidelines. In order to adjust medical guidelines, you need convincing positive results to change what's already being done. And the reason for this is it stems from the Socratic oath that are taken by physicians: do no harm. You don't want to take a risk with a patient when you already know you can help them a little bit. Okay, not as far as you want to, but you certainly are helping them with their current therapies. And so this means that ideas cannot be acted on easily, even if basic science or bench science says that a change will help. An example of that is it's pretty clear from basic science that starting nucleoside analog therapy for hepatitis B virus is very likely going to eventually reduce cancer incidence. But of course, who's got 40 years to go check that out? And so you have to be conservative here. And I don't intend to be coming across as being critical of this. You have to do that. It's important because the number one goal is to protect the health of the patients. Okay, so just summaries and a few final thoughts here. HBV and HDV are very, very different. Although hepatitis delta always comes associated with HBV, they are very different beasts. Curing HBV will cure HDV, although not necessarily immediately. You have to wait until the cells that are replicating hepatitis D virus are killed by the immune system. The therapeutic goal for current drug design for both HBV and HDV is something called a functional cure, which is biologically defined as no detectable viral replication and no disease progression after treatment stops. That's not the clinical definition, it's the biological definition. Finally, HBV is a very complicated virus and has many potential drug targets that drug designers can hit. HBV drug development, consequently, is vigorous against many targets as money has flown into drug discovery for HBV over the last 5-8 years. Unfortunately, hepatitis delta virus is much simpler. That means many fewer targets to try and hit. And consequently, HDV drug development, to tell you the truth, is really quite minimal. Underinvestment in HBV and HDV, the entire spectrum of primary basic research all the way to clinical development for decades has slowed drug development, and developing new drugs for HBV and HDV is influenced by the current state of care for the viruses, and that must be taken into account anytime you're considering how things are moving forward. And with that, I do believe I am done with my presentation, and I will terminate the sharing.
Wonderful. Thank you so much, Dr. Tavis, for a really comprehensive overview of current HBV and HDV therapies and for highlighting some of the really real challenges and areas of hope as well. So just a reminder, I see there's already some questions in the Q&A box. Thank you so much for that. And if you have any further questions, please feel free to put them there and we will address them at the conclusion of all three speakers' presentations. And I will now, I'm very excited to introduce our final speaker. We have Dr. Graham joining us. So Dr. Cami Graham is an assistant professor of medicine and a staff physician in the Division of Infectious Disease at Beth Israel Deaconess Medical Center in Boston. And she's also the medical director of Project RISE. She's the founder and co-director of the Viral Hepatitis Center in the Division of Infectious Disease and has worked to design hepatitis B and C treatment protocols, and also to train fellows in the management of HIV, hepatitis B, and hepatitis C, along with other infectious diseases. And at Project RISE, she provides medical oversight to a harm reduction organization that provides STI screening and treatment and syringe exchange services. So Dr. Graham will be speaking to us today about data interpretation and how to be a more informed consumer of scientific news. So Dr. Graham, I will now pass it over to you. Thank you so much.
All right. Let me share my screen. I'll wait for someone to stop sharing it. All right. All right, does that all look okay?
Yep, looks great.
Okay, so what I wanted to do today was kind of bring you into my head when I think about how am I going to get the most out of this medical conference that I'm about to go to possible. And a lot of what it has to do is the preparation to get ready to go to a medical conference. I think what I'm going to talk about is going to be applicable to any medical conference that you might go to. It's actually applicable to getting your hands around all of the data that start to flesh out what you just heard from Dr. Tavis in terms of the complexity of this disease. My disclosures, I don't have any disclosures for Hep B. I do receive royalties from Up to Date for a Hep C section, and I work at PureTech. And I'm going to tell you a little bit about each of these hats because they do inform my perspectives and what I want to be able to tell you about today. So this is me showing my favorite mask, and I've got my favorite candle of Tony Fauci here. And so this first hat I wear is as an infectious disease physician at Beth Israel Deaconess. And I've spent about 20 years, probably more than that, trying to educate my colleagues, other physicians at Harvard, on hepatitis B. And this is a remarkably difficult virus and disease to understand. So if you feel confused at any point about what you're hearing around hepatitis B, you've got lots and lots of company. I think the downside of how complex it is that you may actually know more, and you may be advocating for patients who then know more than their actual physicians that are taking care of them. That is a substantial risk. So the second hat I wear is I'm medical director at Program RISE. This has a syringe exchange program, STI screening and treatment. This is not a medical center. And so we are beginning to integrate medical care into this center, because we realize it's where people come, where they feel safe, where they feel comfortable. They aren't going to medical centers. And so we have about 100 people on prep right now, and we're starting a hepatitis C treatment program. And then I'm vice president of medical affairs at a company called PureTech that has no liver-related products. I've gone in and out of pharma for the last 15 years. There's pluses and minuses of both pharma and academic medicine. So I'll spend some time in one, and then I'll wander over to the other. And now I'm back in pharma. But that allows me to understand how pharma works. And when you look at that list of drugs that are being developed at the Hepatitis B Foundation that Dr. Tavis just showed you the link for, and it's an excellent website. But I think all or almost all of those drugs are being developed by the pharmaceutical companies. It's because it's really hard to develop a drug, and you need a huge number of people with a lot of different expertises that do not exist in any one academic center or government agency. Oh, and I want to just tell you, there's another hat that I wear. That comes from being an AIDS activist in the late 80s. I was in ACT UP, and I was on the data subcommittee of ACT UP, and I worked with other ACT UP folks to review data, to review protocols. And I was armed at that time with a bachelor's in biology. And I remember how complicated it was to understand the terminology, and I spent a lot of time. So I'm remembering that period of my life when I think about you all now. Some of you maybe haven't been trained in higher sciences. You're armed with courses in college. You can do this, and you can make an amazing impact for patients, and it's hard. Okay, so I'm going to talk a little bit about the homework that you can do, again, to get the most out of a medical conference, or just get the most out of work that you are planning to do around understanding the data around hepatitis B drugs. So the first thing is to think about the hats that you're wearing. Are you somebody that's living with the condition? Today we're talking about hepatitis B and Delta hepatitis, or do you have close family, close friends with this condition where you're really trying to help them or yourself understand personal care choices? And I will say, I first became really passionate about understanding hepatitis B because my father has chronic hepatitis B. And that's a really important connection that might drive your focus and your energy. Are you a peer counselor? Are you trying to translate really complicated data into understandable information that's actionable to that person that you're working with? Are you an advocate for guidelines or policies? Are you a blogger or influencer? Maybe you have all of those hats on. This is going to impact how you think about approaching the data that you want to master. You're going to map out the topics to cover, and I'll go over that a little bit. And then the main thing I want to spend time on is researching existing data in preparation for the conference, because this is actually not a straightforward task. Okay, when you go to a conference, broad topics typically are grouped together. It might be a section of posters, 850 to 865 are all on preclinical data. Or it might be a particular oral session. We're going to hear all the talks on later stage drug development or natural history. And so think about what are the couple of things, maybe pick like 3 from this list that you really want to spend time digging into. I would suggest that you're probably going to spend most of your time on phase 2B and phase 3 if you're focusing on drug development. Very early trials, I think you will start to get very familiar with the graveyard of failed drugs that haven't got past phase 2A. But in any case, sometimes it is good to kind of see sort of what's coming down the pike. Phase 4 and post-approval studies tend to actually be really important. It helps you understand how that migration is occurring from the very sort of narrow population that's studied in your phase 1, phase 2, phase 3 to now what actually happens in the real world in a broader, a more relevant audience, especially to maybe people that you're working with. Don't forget about health economics. Patient-reported outcomes has become increasingly important in drug design. Are we actually making people feel better? And then you may be either looking for studies that incorporate the patient voice in the drug design, or you're looking at studies and you're saying, wow, it doesn't look like you ever talked to a real life human being. And you need to actually listen to people with lived experience. And so I'm going to have a strategy to figure out how to incorporate that. I'm actually really interested in diagnostics. I'll mention that in a little bit. And then don't forget the really important thing you do at a conference is networking with people. And every single time you go to a session or a poster session, it's also an opportunity to network with people that you want to maybe either just collaborate with or share strategies, something like that. Okay, so now I've got my pharmaceutical hat on. How does a pharma company actually share information and data? Because almost all of these drugs are being developed by the pharmaceutical companies. The one that you're going to be probably most familiar with is your medical journals. And so I'm not going to spend a lot of time on that, except for don't forget the supplement. Sometimes the most interesting nuggets of information are found in the supplement, and often it's kind of buried. It's like a link that you have to click either within the text or on the side of your journal banner. So maybe you want to find out, well, what happens in subpopulations, people over the age of 65 or people in rural areas, whatever you're interested in. Sometimes you'll find it there. And sometimes you can find the full protocol. How representative is this study to the people I actually am working with, representing in the real world? What we're talking about today is getting ready for ASLD. So conference presentations. So oral presentations, those are your big symposia where they're going to go through probably about a 10-minute presentation. I'm going to give a shout out now to Jules's NATAP site. He has an excellent relationship with almost all of these companies, and will often have the full slide deck that he posts soon after that presentation. So you don't have to surreptitiously try to, take pictures if he happens to actually post these slides. And then posters actually are a unique, different opportunity. They might include secondary analysis, patient-reported outcomes often end up in posters, but they're a chance to really look hard at the data, and they're a chance to talk with the author. Usually like-minded people are all clustered around the same poster, so it's a chance to find other people who might have similar objective to you. Now, press releases. You know that drug companies release lots of data in press releases. Why do they do that? It's because if they're publicly traded, data that can affect the share price of the company is called material. And material data has to be held incredibly closely, because if it starts to leak out, then people could trade on it, and that's insider trading, and that's illegal. So when a company has locked its database, there's a tiny number of people, it might only be 4 or 5 people in the entire company, are looking at those data, and they are deciding what it means, and they're deciding what the top line message is going to be and what the top line data are going to be presented. And when that data gets released, that's usually around the time the entire rest of the company is informed. So, you know, if you say to like somebody you know in a drug company, hey, why didn't you ever tell me about blah, blah, blah? They very well had no idea until 10 minutes before you did. And then investor presentations and filings. In most companies, you can actually find the entire corporate slide deck, and that might give you a flavor of what that company thinks they're doing, what their long-term plan is. And then your 10K and 10Q annual and quarterly filings often have quite a bit of information in there. If for some reason you feel like you really need to drill down into what's going on with that drug and what's the likelihood it's going to actually make it through the full development process. Okay, so here is an example of an important study in hepatitis B called Retract B, where they took people on nukes and then took them off after a period of time and watched to see what happened. And the point I want to make is that data from this study that was officially presented in an important journal, Gastroenterology, in 2022, were presented at ASLD in 2020, EASL 21, and ASLD 21. So if you really wanted to know about this data, you were following this, you would be following the data coming out of the conferences, and then that manuscript really kind of is tying it all together, and you know, and often it's your final data, because sometimes early data might need to change a little bit as you keep doing the analyses. Okay, so now you're getting ready to go to this conference. You have to start somewhere. You're going to start gathering the data that you need in order to be able to effectively analyze the data at the conference. Because if you go with no plan, you're going to be completely overwhelmed, and you're not going to know whether you're coming or going. So the first thing that you need to do is develop that initial list of hepatitis B drugs in development that you're interested in. You can go to webinars like this. You can go to NATAP, Hepatitis B Foundation, other trusted websites, to get that sort of that initial list and that initial slice of, this is what I want to focus on. You can go to review articles. I will tell you, probably by the time they get published, they're out of date. But they might help you with sort of that overall, this is, you know, kind of like Dr. Tavis just did, these are the classes, this is sort of how these things are going to work together. Okay. The next thing I do, once I kind of get my list of drugs I'm curious about, is I go and check the status of the studies on clinicaltrials.gov. Is this study recruiting? Is it a phase 2 study? Does it look like it's an early phase, later stage? What are the inclusion and exclusion criteria? Who are they going to be looking at? Are these only e antigen negative patients? Is there a certain age group that they're focused on? And then confirm those development plans using the company website, press releases, investor updates. And I'm going to show you a couple of examples. And what you're going to find, if you go to the Hepatitis B Foundation website, you're going to find, like, I don't know, it's like 50 or more, maybe 100. It's an overwhelming number of drugs that are in development. You can't possibly keep track of all of them unless you're someone like Jules. But what you can do is have a few that you're really interested in, and then divide and conquer. You find your colleagues, join listserv, share your tactics. You know, you cover these, I'll cover these, because you've got some common. You create a simple to understand presentation for people who are trying to understand the role of research in their lives, something like that. Okay, so I'm going to give you an example of a drug that I was very curious about for a while. It's one of the core inhibitors, capsid inhibitors, the bivprevir, and it's a first generation. It was just being presented at EASL, the European Association of Study of Liver Disease, just a few months ago in late June. And this was a great study. It showed that when you combine this core inhibitor with entecavir, you got better efficacy than entecavir alone. And you can see here that there were 4 different posters on this drug. So looks pretty promising. And I want to point out here, this is true. These posters will be available subsequently on the events and presentations page in the investor section of the company's, it's Assembly Bio's website. So it actually takes a couple of more clicks than what's described there. But if you click through, if you wanted to know the actual full poster, not just like a couple of slides, you can get the full poster, and that's pretty common at many drug companies. Okay, but before you go digging into the website and looking at all of the posters, go and look at the press releases. And here are press releases. This is just from Yahoo Finance for Assembly. And what you see here is that they discontinued the clinical development of this drug. Okay, well, now there's no point looking at all those posters because they've discontinued the clinical development. But you may remember this was an interesting situation where this small company, Assembly, partnered with another small company focused on hepatitis B drug development, Arbutus, to create a combined trial. And this is really important because I think all of us suspect that, like we saw in HIV and like we saw in hepatitis C, we are probably going to need combination therapy. And there's a tendency of companies to want to have all their own toys in their own company. But no one company might have all of the right elements. And so we want to see partnerships. And this was a partnership that had happened. And so what was Arbutus doing? Go to their press releases. And you can see here, they have a press release that's it's a little bit ambiguous, but it kind of sounded to me like, yeah, we don't care. We're going to continue our phase 2 study using this Assembly drug in combination with our investigational drug. Okay, so now you know that it's continuing to have a life in this other realm, and that'll be sort of interesting to see what happens there. Okay, but maybe you're now going to focus on, you know, are there going to be any data with AB 729 at AASLD? Okay, so here's another example. This is Ionis. They have a press release of positive phase 2B data. Now, this is important because I just mentioned there's a graveyard of failed drugs in early development. And so something getting through phase 2B is really important because the next phase is phase 3. That's your pivotal trial. That's where you basically are laying out the data that gets presented to the FDA for potential approval. And so this is a really exciting milestone. And this is a drug that actually was made by Ionis, but it's partnered with GSK. And so let's look at this press release here. You can see, first of all, that they're now planning to go on to phase 3. So positive enough data that they're going to go into phase 3. So, and they're going to go in as monotherapy. Now, I already said, I think we're expecting that many of the drugs that we're going to, of the regimens that we're looking at, are probably going to be combination. So, so that's interesting that they're going to go in with monotherapy. This is actually a really interesting and complicated drug, and it has multiple mechanisms. So maybe in one drug, it actually almost acts like multiple drugs and multiple mechanisms. But then what you also see is that GSK is exploring potential combination treatments. Okay, so let's see what GSK is doing. This is where we're going to go back to clinicaltrials.gov. So you can search for, you can search for the name. Bepirovirsen. I have no idea how to pronounce it because I only read it. I don't actually say it. And so we've got one study with that drug, and it's in combination with another GSK drug. And then you've got another study that comes up that you can see on the drug side is now using basically the GSK number. So the number is 8836. And it's being combined with a different GSK drug, some sort of biologic, and you see that it's a hepatitis B targeted immunotherapy. So when you go to clinicaltrials.gov, you have to then sort out what exactly is it that's being studied, because the same drug might not be described in the same way in a series of clinical trials, even done by the same company. And so this particular first one, when you include healthy volunteers, that means it's a phase 1 study. And when you have people who are actually affected by the disease, you can call it a phase 1B, phase 2A. But it's an early study. These tend to be pretty small. Just giving you a hint, what's going on with this drug? Is it safe? Does it seem to be safe so far? Okay, and then once you start getting into these larger studies, I happen to look at this one, it's considered a phase 2 study. This is what I call aliases. And so as you're looking through your clinicaltrials.gov, press releases, manuscripts, posters, companies may be using different designations for each of their drugs. And so making yourself kind of a cheat sheet, when I want to see 8836, I know it can be described in one of 4 ways. And when I see one of these 4 ways of seeing this drug, I'm going to know it's that one drug that I'm trying to follow. Same with 5139. This is an inhibitor of these human enzymes that are involved with hepatitis B metabolism, kind of an interesting combination approach. And then 8869 is a 3-component therapeutic vaccine. And so it's sometimes described as 8869. It's sometimes described as 3-component therapeutic vaccine. And it's obviously described here in this clinical trial as chronic hepatitis B targeted immunotherapy. So again, if you can kind of create a little cheat sheet to sort this out, it's going to make things a lot easier because you never know which one of these is going to pop up on a poster. And you might think that you're seeing some new data when it's really another alias being used for one that you're already tracking. Okay, so I'm gonna kind of come up with another scenario here, which is, let's say you are talking to people living with hepatitis B who are currently either considering starting antiviral therapy, or they are on antiviral therapy and they just want to kind of know, is this something I'm going to have to take for the rest of my life? Like, why am I taking this? And you may want to say to yourself, well, what are the criteria for people to get into these new potential functional curative therapy clinical trials? Because if this is who they're letting into the clinical trial, then that's probably what's going to end up on the product label, although things I will say can really change. And for whatever reason, right now, they think this is the right group of people to maximally demonstrate benefit, if there is benefit to be had with these drugs. So in order to kind of answer that question, what's the value of being on therapy, of being on suppressive therapy? I just randomly grabbed a handful of currently enrolling hepatitis B studies and asked myself, are they requiring that people be on nukes? And if so, how long? And do they have any requirements for people being virally suppressed? And you can see here that there's 3 studies that require that you've been on stable, either entecavir or one of the tenafavirs therapies for at least 12 months, and then some for a shorter period of time. Many of them want you to be suppressed. They've got some different definitions, different time periods. A couple specifically point out at least 6 months of viral suppression. Others don't specify a duration. There's one that's this Hep T cell that's an immunotherapy that actually does not want you to be fully suppressed. I think because they're looking to see if this therapeutic vaccine can actually create suppression. Okay, and this is my last slide, and this is some practical advice. And this is having gone to medical conferences for, I don't know, 25 years or something, and just each time honing a little bit better how to keep myself from being completely frazzled and confused. So one thing I do is I write down a little schedule each day. You can download a schedule of events in your phone. You can print things out. but just having a little quick cheat sheet that allows you to see, oh, this oral presentation is in this part of the conference building, but then I have to run like half a mile, over to the complete other side of the conference center to get to this other one. I'm going to need to leave myself extra time for that. Sometimes when people first start going to conferences, they think, oh, I'm going to go to this talk over here, and then I'm going to run over to this talk over here, and then I'm going to go to this talk over here. This never, ever works. I allow myself one transfer. So the most I might say, I'm going to start at this oral session for these 4 talks, and then this 5th one, I'm not so interested. I'm going to race over to this other conference room, and I'm going to listen to the second half of this. You may not get in that room, especially with COVID restrictions. You may find that you're in a line or that you're watching a TV somewhere. And so I think, when I get to ESOD, I'll be sort of looking at the dynamics and sort of some of that, but that's what I mean by logistics. You don't necessarily, let's say what you're talking about, the posters, you're going to go see the posters. You can usually, like I said, they're grouped. And so you can say, well, I'm going to look at 850 to 865 because those are all the posters that are kind of covering, the area I'm really interested in. And I like to get to the posters as soon as they're hung up, when I can, take a look at them, because I'm thinking about questions for that author when they've got that hour or so that they're supposed to stand next to their poster and present it. Of course, you might have your list of people that you want to look for. The exhibition booths, they always have, they often have 2 sides. If they've got a commercial product, then one side is their commercial side. And these people are trying to sell drugs, and they actually are not particularly helpful for what you're going to be wanting to ask. There's a medical side. It tends to be physically separated from the commercial side. So if you want to find out more about that drug in development, can you send me additional information? Whatever your question is, you go to the medical side for that. If you say, wow, your company really is not exhibiting a good understanding of what it's like to live with this disease, I would like to talk to people about that patient perspective, you go to the medical side. We talked about keeping a chart of your drug aliases. The lines for a bottle of water or a cup of coffee are ridiculously long and ridiculously expensive. So I always draggle around a snack and a bottle of water. And then you'll probably get more out of a nice dinner with colleagues in the evening than attending an industry-sponsored event. Okay, that was my presentation. Let me stop my slides.
Thank you so much, Cami. You were kind of taking me down a personal trip down memory lane to the heyday of hepatitis C drug development, and listening into investor calls and reading the quarterly filings and all of that stuff. So we have a few minutes left, and thank you for people who've submitted questions in the Q&A box. I want to start with an kind of overarching question. We started, and I want to remind people that we do have a second part to this webinar coming up on the 19th, 2 weeks from today, with a discussion about some of FDA's activities around engaging partners and looking at equity and diversity in clinical trials and drug development. And just so that we have a kind of nice foundation, putting this out to everyone, maybe Christine, if you'd like to start, what is the foundation for the rationale for making sure that as we look at these clinical trials for new hepatitis B agents and combination therapy, or really any kind of drug development, that we're keeping equity and diversity at the forefront?
Sure, Dana, that's a great question. So, diverse representation in clinical trials is not just a simple matter of biology, but it's a matter of health equity, fairness, and public trust. Equity in clinical trials is particularly important when we think about access, equal access, and accrual in clinical trials. Underrepresentation of minority patients in clinical trials really compromises the generalizability of trial results, could lead to miscalculation of disease-free survival rates, and also, would affect treatment efficacy understanding too. So it really is quite important to have diverse representation within our clinical trials.
Thank you. John or Cami, anything you'd like to add to that?
If you don't know how the drug is performing in their diverse population, you can't make the right treatment decisions.
And I'll just add, you can say you need patient voices in here. You need to be listening to people as you're designing your clinical trials. I think more and more companies are moving to that direction, but especially some of the smaller companies, it might not occur to them. They might not have the people in there that really understand that and understand the value of incorporating people with lived experience into their study design process. So advocates can make a really big difference actually in helping companies understand how to do better with this.
Thank you all. I want to go to a question that from our audience, a couple of questions that Joe put in the chat about hepatitis Delta in particular. Do we have efficacy data on Hepcludex in patients with advanced cirrhosis? And what should we be thinking about as we talk about this concept of functional cure? John, I think that you offered a lot of great insights that curing hepatitis B would ultimately result in the clearance of Delta because Delta is dependent on hepatitis B viral replication. Does that extend to a functional cure for hepatitis B as well?
Yes, with one caveat is that it is possible that her surface antigen being produced from integrated copies of the HBV genome could support spread of Delta within that cluster of cells that had a particular functional integration. So there is a complexity there, but eventually the immune system would win by attacking cells expressing the Delta antigen. At least that's the current belief.
Thank you. And in terms of, and in terms of indications for Hepcludex, I know it's not approved in the United States yet pending FDA review, but do we see indications of benefit for patients with advanced cirrhosis?
That I don't know that level of detail. I just don't know that level of detail with it. Sorry, I can't answer that one.
Okay, thank you. And a question from Prince that came up, who is talking about the experience of being a hepatitis B patient from Nigeria and not having easy access to a viral load. And so working on other diagnostics like HBe antigen and trying to, but not falling under that category for which current hepatitis B treatment guidelines would indicate treatment. You referenced this in talking about some of the hurdles around treatment indications and perhaps conservative treatment guidelines about people for whom treatment is warranted based on an assessment of risk and benefit. Do you have a broader, maybe editorial take on your thinking about are the current indications for hepatitis B treatment appropriate, too narrow, should be reconsidered, and what are the challenges in thinking those questions through from a guidelines perspective?
Did you want me to take that one?
Yeah.
Yeah, now this is a personal opinion. Remember, I am not a physician. I never have the responsibility of sitting in a exam room with a patient. So take it from the perspective of a virologist who studied the virus for his entire career. Is yes, I believe the treatments are too conservative. I believe we should start treating very early, like we do with HIV. At least with very, very safe nucleotides. This, of course, requires doing this only in patients that are not contraindicated with any aspect of the drug. But the reason is the virus integrates very rapidly once it gets into a cell, and we know those integrations can be oncogenic to some degree. There's still arguments over how oncogenic they are, but they can contribute to forming of cancer. So I personally believe that suppressing the burden of integrated DNA is quite important, and starting nuke therapy right away as soon as a person is diagnosed would do that.
Can I add a
and-
different
oh, sorry,
perspective?
go ahead, Cami. Yeah.
Right, so a completely different perspective that gets to the same answer is the current guidelines are complicated. There are not enough ID experts who are comfortable with hepatitis B and hepatologists to actually take care of everybody. Care needs to be with primary care and family medicine and perhaps at places like Program Rise. This is where we're going to actually impact this disease. And the more complicated it is, and the more complicated testing algorithms, the less likely it is that people are going to receive a treatment that can decrease the risk of cancer and cirrhosis. And I think in terms of resource limited settings, in many ways at the Syringe Exchange program I'm with, I kind of feel like I've got resources. I'm hustling for grants all the time, to be able to do the testing that we need. And we don't have enough funding. And so we end up making decisions the best we can based on with limited resources. And, so I think there's a whole line of people who are working on how do we actually simplify this, not just for places like the US, but for the rest of the world.
Great, thank you so much. And I'll just reiterate that these aren't intended as clinical advice so much as a broader perspective on where we are in treating, curing, and hopefully ultimately eliminating hepatitis B and Delta. This takes us up and a little past our allotted time. And on behalf of NVHR, I want to thank our three wonderful presenters, Dr. Lee, Dr. Tavis, Dr. Graham, and express my gratitude to Beatrice and the Hepatitis B Foundation, our partners in this. Before I turn it back to Beatrice for closing words, I'll flag that my colleague Adrian posted the registration link for part 2 of this webinar, as well as a link to NATAP, which Cami mentioned, lots of helpful write-ups, reviews, access to latest data and clinical trials. So Beatrice, do you want to close us off?
Yes, absolutely. Thank you so much. Adrian, would you mind just sharing the last slide, please? Thank you so much. So just echoing Daniel, we really want to thank all of our speakers and to thank you for everyone for participating in today's webinar. We really hope that you found it informative and valuable to your work or your life, and that you can apply some of this information moving forward, whether you're attending the upcoming AASLD meeting or in other sort of areas. A recording of this webinar and slide presentations will be emailed out to all attendees. And for more information about the Hepatitis B Foundation and NVHR, including the drug watch page that was mentioned a few times, you can feel free to visit our website and or reach out via email using the information listed on the slide. And then just a reminder, as Daniel said and Adrian shared, there is a link to part 2 of this webinar, a series which will be focused on more of the Hep B and V clinical trial experience as it relates to access and equity. And that's going to be 2 weeks from today on Wednesday, October 19th at 2 p.m. Eastern time again. So please register for that and check out the other resources. And thank you so much again for attending, and have a wonderful rest of the day.
Thank you. Bye, everyone.
Thank you. Bye.